Hello and welcome to another class of ABM Sciences, and I am Abhishek with you. So last day I have given title about the virus that can act against Superbug. So I have not explained that thing properly. So today I'll be telling you about what is Superbug and how this virus is helping us against fighting these bugs things. So what is a Superbug? So a superbug is a bacteria. But why a bacteria called as a superbug? Because it has some characteristics. Because that's why it is called a superbug. So what are the characteristics? Antibiotic resistant. Resistance. Now, a bacteria can be resistant to one antibiotic, two antibiotics, three antibiotics, and multiple antibiotics. So, multi-drug resistant bacteria. So, when a bacteria is resistant to one or two bacteria that are commonly used, actually these antibiotics against which the bacteria is resistant are commonly used antibiotics. Commonly used antibiotics. Suppose suffering from diarrhea, okay, that causes due to Salmonella typhi. So if I am attacked by Salmonella typhi, the doctor will prescribe some of the antibiotics. But if the strain which is present inside me and which is causing the disease inside me, that strain is particularly resistant to, suppose the antibiotic is amoxicillin. So amoxicillin is the commonly used antibiotic against gram-negative bacteria. So now doctor will be prescribing amoxicillin to me. I will be taking the medicines for 3 or 5 days according to the dosage indication, and after the 5 days the bacteria will be there because it has no effect. The antibiotic has no effect on that bacteria because the bacteria is drug resistant. So if I went to the doctor again and check my bacterial level again with the different diagnosis method, then doctor will prescribe me 2 to 3 combination of antibiotics, okay, so that the bacteria can be eradicated from my body. So that means the bacteria is resistant to one antibiotic. If the doctor prescribes me 2 to 3 combination of antibiotics and even after that the bacteria reside within me and without any complications, with complications, it is present with me and it causing a life-threatening disease now, so that is called a superbug. So if it is multi-drug resistant, then it's called superbug of superbugs, okay. So that's why the superbug is a dangerous thing in case of bacteria, because the multi-drug resistant bacteria have no answers to medical science. Medical science has no answers to multi-drug resistant bacteria. That's why superbug is such a dangerous thing. Now, as antibiotic is not responding to them, what will happen? We need to go for another alternative method, and that is bacteriophage. So you may ask that bacteria is so rapidly evolving itself, means they are resistant to antibiotics. So why not the bacteria is resistant to bacteriophage? It is also a kind of, you are telling as a drug or dose or something. But here I will say that there are few characteristics of bacteriophages which is enabling the bacteriophage to fight against this superbug again and again, no matter how many times you will treat them. So what are the characteristics of bacteriophages which makes them as a potential fighter for superbugs? So that is, they are very specific, right? So they are very specific to their target organism. If it is Salmonella typhi, it will be only used against Salmonella typhi, not only Staphylococcus aureus like that or E. coli. So it's a species specific, okay. Next is, it's a live and rapidly evolving agent. So likewise bacteria, the bacteria is evolving themselves rapidly against the antibiotics, but we cannot do anything there. But here the bacteriophage is also evolving themselves against the bacterial modifications. So there will be a continuous fight between the bacteriophage and the bacteria. So these are the second advantage. It is easily available. You can collect or isolate bacteriophages very easily from wastewater treatments or several water bodies. Cost effective. There are several experimental or clinical trials happened in the Russian or Georgian countries where they have successfully applied bacteriophage cocktail, actually mixture of different bacteriophages for the same or a particular bacterial pathogen, where they have killed or successfully suppose they have 70 to 80% successful rate when they are using this bacteriophage cocktail against a particular antigen or particular pathogen. So that's why it's a very cost effective and no adverse effect on humans till now. So this is a huge advantage against antibiotics to the bacteriophages, because whenever you are consuming antibiotics, it will clear your normal flora. So it is enabling the pathogen to come and sit over me and change everything and make me diseased. But in case of bacteriophages, they are not harming. It is experimentally proved with different research findings that bacteriophages are not going to harm the internal flora of the human beings. It is, as it is, I told you, it is very much specific. It will not act on other bacteria. It will act certainly specifically to the pathogen, that is our superbug. That's why the virus is so efficient in fighting a superbug. So I hope it's clear now. If you have any questions, please ask me. I'll be telling you some terms that will be familiar with in future, and that will help you in your exams also. So one by one I'll be going to that. So first one is lysogen. So what is a lysogen? Lysogen is the bacteria. Lysogen is the bacteria which is able to harbor a bacteriophage. So lysogen is the bacteria which is able to harbor a bacteriophage. It is called lysogen. Now what is lysogenic conversion? Lysogenic conversion is when a bacteriophage infects a bacteria, that time their cellular surface, the structure of the cell wall, some of the proteins or the lipids or the arrangement around the cell wall gets changed due to the infection process of bacteriophages, and that is occurring due to the effect of the bacteriophage, that is the responsible infecting organism. That's why the conversion that happened to the bacteria due to the bacteriophage is called lysogenic conversion. So to the bacteria due to bacteriophage. These are the few terms. Now, two types of phage are there. Okay, two types of phage. That is, one is virulent, one is temperate. What is virulent and what is temperate? So virulent phages will be going through lysis. Means when the bacteriophage is infecting the bacteria, that time bacteriophage will grow inside it, will multiply, and from millions of copies, and that copies will lyse the cells, and then it will be out of the cells. So there will be no host after lysis of the host organism. That's why it is a virulent phage, means total destruction of host cells. Okay. Now next is temperate phage. Temperate phage is totally opposite of the lytic phage or lysis. That is, here the bacteriophage genome can be integrated in the bacterial chromosome. Suppose this is the bacterial chromosome. This is the bacterial chromosome. Now the bacteriophage now this blue color is the bacteriophage genome. So what are you able to see here? That the bacterial chromosome now harboring a bacteriophage genome. That means that now bacteriophage genome is integrated into the bacterial chromosome. This condition is called prophage. And this type of bacteriophages which are able to integrate their genome into the bacterial chromosomes are called temperate phages. So what happened in this kind of amplification is this: there will be no lysis, no virion formation, only division of phage genome. So in this case, in case of temperate phages, the bacteriophage genome will be dividing with same copy number or same amount compared to the bacterial genome, because it is integrated. So whenever the bacterial genome is going to duplicate itself, it will be also duplicating the phage genome. That's why it will be replicating to different billions of bacteria. Now it is there in the billions of bacteria. Now if somehow there is an adverse condition, or the bacteria are induced by some kind of nutrition deprivation or UV treatment or some kind of treatment, drugs, anything, if you induce the bacteria, that time what will happen, this phage genome will be cutting out, means it will get out from this bacterial chromosome and it will go to lysis. So understood? Whenever the bacteria will have an adverse situation, that can be any kind of nutritional stress or UV treatment or any kind of drug treatment, that time may the bacteriophage, that will induce the bacteriophage genome to getting out from the bacterial genome, and then this bacteriophage genome will act as an lytic phage, means it will replicate itself, it will form the virion capsid heads, tails, tubes, seeds, everything will be prepared. A fully formed virions will be started reading inside the bacteria, and at last it will burst the cell. So that may also happen. So this is called temperate phage. Now these terms can also be called as lytic phage, and this is called lysogenic. So the phages which are able to lyse the bacterial cells and always lyse the cells is called lytic phages, and the phages which are able to integrate their genome inside the bacterial chromosome are called temperate phages or lysogenic phages. So these are two domains of bacteriophages which will be acting on bacteria depending on their nature. Now whatever phage, what the structure I have seen you last day, I have shown you, that is T2, T4, T5, T6, T7, this kind of bacteriophages are present there. Okay, sorry, this is T7. Now the odd numbers and even numbers of bacteriophages. So these bacteriophages are dividing depending upon their family. Okay, family like Cephuviridae, Myoviridae, Tectiviridae, different types of families are there depending upon the capsid head, either it is elongated or polygonal in shape, or maybe it can contain, sometimes there is examples of bacteriophages where the bacteriophage contains envelope inside the capsid. So these are exceptional cases, but most of the cases that these T phages, T even or T odd phages, contains a capsid head. Likewise, I showed you in the last class. So that time it will be dividing whatever the process like penetration, absorption, and attachment, and then injection of the chromosomal DNA or their viral DNA. So this is the total process. And one more term I will tell you what is that. That is super infection. So what is super infection? Super infection is the term where if the bacteria is attacked by a certain bacteriophage, after the attack of the bacteriophage, there are certain changes that will occur on the cell wall or the attachment sites of the bacteria, and that changes may not allow the same bacteriophages to attach, or any other type of bacteriophages to attach to the same cell. This is called super infections. Means they are immune to any kind of further infection through the same bacteriophages or any kind of other bacteriophages. These are called super infection. So if you have any problem in understanding any of my description here, you can ask me questions in the comment section and I will answer your questions. So I will be telling you again what is superinfection. Superinfection is the changes that occur. I will be writing here so it will be easy for you. So changes that occur due to phage infection and as a result the same phage is not able to infect again. So this is called superinfection. This has been seen in some of the bacteriophages like epsilon phages. So here are some examples of the terms which are commonly encountered during when you are studying bacteriophages. So I will be giving you some examples like these are not examples, these are exceptions actually. So like PRD1 phage. So this is an exceptional phage where inside the capsid there is one envelope, and inside that chromosome is present, and here they have some kind of spike proteins through which they attach to the bacterial site of attachment. So these are spikes. This is membrane or envelope, and this is capsid, and this is genome. So this is an exceptional phage where within the capsid there is a presence of a envelope, and inside the envelope there is a chromosome, and the attachment process is also different because they have spikes, and through these spikes they attach to the bacterial cell. And there the spike is gone, and then they inject their chromosome or the genetic material into the bacterial cell, and this is the process of this kind of phages. So this is exceptional case, so we will not be going in detail with that. So I will be telling you about the common phages and common process of replication like T4 phages. This is bacteriophage replication and protein synthesis. Here we will be learning about how the bacteriophage is multiplying their copy number or their genome inside the bacteria with the help of bacterial machinery. Okay, so much modifications is required for bacteria during bacterial replication and protein synthesis. But before going on that, I want to tell you that within the capsid I told you that there is the nucleic acid. See, so this nucleic acid can be of what? DNA or RNA, right? So now in DNA there may be present single-stranded DNA or double-stranded DNA, or in case of RNA there may be single-stranded RNA or maybe double-stranded RNA, right? So within a bacteriophage capsid there may be first either DNA or RNA. Now if there is DNA, it may be a single-stranded DNA or maybe a double-stranded DNA. If there is RNA instead of DNA, then it may be single-stranded RNA or double-stranded RNA. So there is example for everything. There are bacteriophages present from all the 4 domains. So their process of replication is differing from one to another. Obviously, because the single-stranded DNA genome and double-stranded DNA genome, there is a difference between their replication process. And in case of RNA, this is totally new form may be formed. So this is the scenario of DNA and RNA. So the replication process in protein synthesis is a long-term process, and this class is going to be very, very long. So I thought I will be describing you in the, I think 2 more classes will be there for replication and protein synthesis, 1 class, and one will be how the bacteriophages will choose, or they are choosing either to go for lytic cycle or go for the lysogenic cycle, either to lyse the cells immediately or being as part of the cells after cells and billions of cells, and then get out of the cells whenever they need. So I think 2 more classes will be necessary for this understanding of the bacteriophage replication. So I am stopping here now, and if you have any questions or any query, please ask me in the comment sections, and do subscribe if you think that this video is good enough for your understanding. Please subscribe. Thank you.